all of your proteins can do double duty and act as buffers. Almost all of them have some buffering abilities, from either a little bit to a lot, just because of their nature of the way the amino acid residues are structured. But this carbonic acid bicarbonate is one of the most important, especially extracellular buffers. The protein buffers, mostly intracellular buffers. This carbonic acid bicarbonate buffer system, extracellular. It's one of the most important ones. The pH of blood tightly regulated between 7.35 and 7.45. If you do not have this memorized, 7.35 to 7.45, do so now. It's a good number to know. Normal range for pH, 7.35 to 7.45. If your pH is below 7.35, that's a bad thing. You are too acidic for your own good. You would be in acidosis. What would put you into alkalosis would be if your pH was above 7.45. Above 7.45, you're too alkaline. Alkalosis. If you're below 7.35, you are in acidosis. You're too acidic. What throws people off, and I understand the confusion, is that if a solution has a pH of 7.2, you're just in a lab room or something like this, has a pH, you just use your little pH meter, has a pH of 7.2. How does that fall on the pH scale?
Neutral.
You're above 7, 7.00000 is neutral. So a pH of 7.2
Is base. Is
would be
alkaline.
basic, right? Correct? Because any number technically above 7 is basic. But if your pH of your blood is 7.2, you are in acidosis. So how can your blood still be alkaline, and yet we say that you are in acidosis? Well, the reason is that 7.2 is below your normal range. Which is why, even though technically it's still on the alkaline side, we still say that you are in acidosis. Most people who are in acidosis never even actually get below 7. That's a pretty severe acidosis if you actually get that far down. But back to our discussion here, is that the pH of blood tightly regulated, and little changes in pH, little changes in pH is bicarbonate because it can take up excess hydrogen ions, reforming carbonic acid. Or if hydrogen ions are lost, then carbonic acid can release hydrogen ions. So this bicarbonate system allows you to deal with conditions that become too acidic or too basic, which makes it a good buffer. And again, the primary source of this carbonic acid and bicarbonate ion is the carbon dioxide in your blood. And the carbon dioxide in your blood, as you already are familiar with, is primarily determined by the rate and depth of your breathing. If you breathe deeper and more rapidly, what's going to happen to your carbon dioxide content? It's going to go down because you're blowing that off. If your respiration slows down, your levels of carbon dioxide are going to go up, and you start to become too acidic. So again, that enzyme carbonic anhydrase is highlighted here that changes carbon dioxide and water into bicarbonate. And the double-sided arrows show you that this is reversible, which means that use your powers of imagination for a second here. I'm going to make up some numbers, and I can do that because I'm a professional. Let's say that for every 1 part of carbon dioxide and 1 part of water, you end up with 1 part of carbonic acid and 1 part of hydrogen ion, 1 part of bicarbonate. So we have a 1 to 1 ratio, right? If we add a lot of carbon dioxide to the solution, follow me with this, all right? We add this end up, this goes up to 10. Well, that's going to shift the equilibrium that way here, right? And so this goes up over here, then eventually that means we're going to convert more into carbonic acid, which means more gets converted into bicarbonate and hydrogen ion, and we shift the equilibrium this way here. If we remove carbon dioxide from the solution, then that's going to shift the equilibrium back in the other direction. So keep in mind that it's always going to sort of, if you shift it one way, it's going to try to even itself out. If you shift it the other way, it's going to try to even itself out. So ventilation pattern, when somebody hyperventilates, the rate and depth of breathing increases. And of course, that means when you hyperventilate, that's going to increase the amount of carbon dioxide that you are blowing off. And if I blow off more carbon dioxide, all right, that's going to cause you to become more alkaline, right? Because you're getting rid of acid, right? Just think of carbon dioxide. If somebody slows down their breathing, for example, this can be an issue when people are on long-term opioids, for example. If you're on a morphine drip, for example, that has an impact on respiratory centers, depresses them. That can send you, if you're hyperventilating, that would send you into a respiratory acidosis. Now, control of respiration. Let's take a look at control of respiration here. The medulla is where respiration is going to be initiated from. The medulla, to repeat, is where respiration is going to be initiated from. And there are two major respiratory groups. There's a dorsal one and a ventral one. The dorsal respiratory group is the one that establishes the basic pattern of breathing. It sends action potentials to the diaphragm. It sends action potentials to the external intercostal muscles. And the way it breathes is that it sends action potentials for about 2 seconds to these muscles. As they contract, you breathe in. As soon as the dorsal respiratory group goes quiet, stops sending signals, what happens? You automatically will breathe out. All right? So when the dorsal respiratory group is active, you will breathe in. As soon as it goes quiet after breathing in, elastic recoil will automatically make you breathe out. Now the ventral respiratory group is only going to be active during forced breathing, especially active expiration. Not during normal quiet breathing, but it's the only one that's going to be involved in expiration, active expiration. The dorsal will always be involved in normal inspiration, but will the dorsal one ever become active during expiration? No. It's quiet during expiration, passive expiration. The dorsal cannot get out when you're coughing or laughing, all those things that are pushing air out of your expiratory reserve volume. That's all from which group? The ventral. So the dorsal respiratory group is only going to be involved in what process? Is only active during what process? Inspiration. Whereas the ventral respiratory group is also going to be involved in what? Expiration. What will dive remind you? That the dorsal respiratory group is only involved in inspiration. But the ventral is involved in expiration, any kind of active process, especially active expiration. So when you're coughing, laughing, in labor, all those things when you are pushing things out. All right, so yeah, nothing more on this. Okay. Other control of respiration is going to come from the pons. Now, the pons will have an influence over the medullary respiratory centers. The pons has no direct control of your breathing muscles. The only thing pons can do is have an impact on the medullary centers. And there are two centers, the anoustic center that provides continuous stimulation for the dorsal respiratory group. Now this might sound a little strange, but keep in mind, as long as the dorsal respiratory group is activated, you will continue to breathe in. All right? So the longer it's stimulated, all right, the slower your respiration rate would be, because you have these long inspirations. That's the anoustic center. Whereas the pneumotaxic center, which inhibits the anoustic ones, all right, stops you from inhaling for long periods of time. And keep in mind, as soon as you stop inhaling, you automatically do what? Exhale. So the pneumotaxic center will speed up your breathing. Pneumotaxic does what? Speeds it up. The anoustic does what? Slows it down. Do either of these control your respiratory muscles directly? Or will they just have an impact on the medullary centers? They affect the medullary centers, the dorsal respiratory group. Now, what will trigger changes in breathing? Well, chemoreceptors that are responding to levels of oxygen, carbon dioxide, and acid, by the way, are some of the more important things that are going to have an impact on it here. These peripheral chemoreceptors are located just adjacent to where we had the baroreceptors that were involved in control of blood pressure, all right? Near the carotid, the aortic bodies there, carotid and aortic bodies here. We have these chemoreceptors that are activated in response to changes in oxygen, or pH, of course. And a decrease in partial pressure of oxygen, or pH, activates them, and that's going to stimulate your dorsal respiratory group, so you start breathing more. Now, the central chemoreceptors are actually located on the surface of the medulla. Keep in mind, the surface of the brain is going to be covered with cerebral spinal fluid, right? That's circulating right outside there. So that's what they're sort of picking up in here. They're activating response primarily in changes of carbon dioxide and cerebral spinal fluid. And as carbon dioxide increases, generally what's going to happen to pH? Carbon dioxide's increasing, pH is going to be decreasing. And those are the things that are going to be triggering breathing. It stimulates the dorsal respiratory group. It's like, you're too acidic, too much carbon dioxide, we need to get rid of these things. And of course, that since changes in partial pressure of carbon dioxide directly affect pH, then small changes in PCO2 tend to have very dynamic effects on respiration. Oxygen is only going to stimulate it if your oxygen levels have plummeted. You've got to have your PO2 below 60. Now remember, normal arterial PO2 is around 100, right? About 100 normal PO2, right? So if your PO2 drops to around 40 or 60 or so, not 40, about 60, that's going to stimulate your breathing. But aside from that, has very little impact on breathing. Most breathing regulated, again, because of carbon dioxide and acid levels. That's the thing that triggers them. So carbon dioxide goes up, it's going to make you breathe faster or slower. If carbon dioxide levels go up. It's gonna make you breathe faster. If your carbon dioxide levels go down too much, what would that do to your breathing? It would slow it down, inhibit it. All right, lastly here, some neural effects here. This is, the baroreceptor reflex is the one that deals with blood pressure. I don't think this really shows up a whole lot, but we mentioned this when we talked about control of blood pressure, that almost anything that would cause an increase in cardiac output, all right, because of lack of oxygen in tissues or things like this, where the heart's beating faster, is almost always going to also increase respiration. So those two things tend to go hand in hand. So if there's an increase in blood pressure, it tends to reduce respiration because it inhibits those centers. Nothing. Last thing I really sort of mention here is that the limbic system, headquartered in your hypothalamus, can also have an impact on breathing, again by its activation of those respiratory centers. Did you touch on this in 109 when you talked about the limbic system and your control of autonomic function? Hopefully you did. So let's sum it up this way. Can your emotional state have an impact on your breathing?
Yes.
Is this done through the limbic system?
Yes.
Does the limbic system itself directly control breathing? No. It will exert its control by affecting the output from what part of the brain? Medulla. Because the medulla is the place that actually initiates breathing. So the pons affects the medulla based on carbon dioxide levels, oxygen levels, pH levels, based on chemical composition of the blood. The limbic system affects it based on emotional state. If you're anxious, your breathing will probably do what? Increase. If you're nice or relaxed and calm, your breathing probably slows down. Last I'll mention here is that do you also have voluntary control over breathing?
Yes.
Everybody agree with that? Can you decide to take a breath?
Yes.
Oh, that's amazing, yes. Which means that there are also going to have to be areas of your primary motor cortex, right? Because that's where all voluntary control is initiated from, correct?
Mm-hmm.
Will your primary motor cortex control your respiratory muscles directly? Not in this case. It's primarily going to affect the respiratory centers again, because again, the medulla is where we're generally going to be initiated from. Okay. Enough. No more questions on this. We move immediately to digestion. So, y'all familiar a little with digestion?
Mm-hmm.
Can your digestive tract absorb polymers?
No.
What has to happen to the polymers in the diet? They have to be broken down by monomers. By what process? Hydrolysis. So, all the enzymes of your digestive system are providing what function? They're all hydrolytic enzymes, that's all I'm trying to get to. All the enzymes are hydrolytic. But when we talk about all the digestive processes, all right, that's just part of the end game here, being able to absorb things here. We start by having to eat something or drink something. What do we call that process of like taking? Mastication is the chewing.
Ingestion.
Just ingestion. Just ingestion. We're just going for the obvious here. Ingestion is just taking in food, eating it here, into the digestive tract. Compulsion, we have to move it along primarily by peristalsis. Have we talked a little bit about that process of peristalsis? Remember that peristalsis is going to involve 2 layers of smooth muscle. Did we have this conversation?
Let's go over
I don't think
So just as a quick reiteration, maybe I didn't cover it very well. Smooth muscle generally has 2 purposes in your body. One purpose is to control the diameter of some type of tube, like an artery or a respiratory passageways. And to control that, you just need one circular layer, which means blood vessels just have a circular layer of smooth muscle around them. Your respiratory passageways to control their diameter just have a what? Circular layer of smooth muscle around them. To propel things by the process of peristalsis, we need 2 layers. We need a circular layer and we need a longitudinal layer. Because the process of peristalsis is very much like the process you use if you pull your pair of sweats out of the dryer and the drawstring has come out a little bit. Never happened to anybody else. Okay, yeah, right. Just me, right. So, any case, if this happened, oh, my favorite sweats, you're not gonna throw them away. You find where that little nub is, it's like buried like 6 inches back, all right? And then you start doing this, right? Like you don't know what I'm talking about, right? You find where the little nub is, you bunch it up, you stretch it back out, right? And then you pinch it off, and you keep doing that bunching and pinching into that little egglet. Is what that little tip, I've learned the name of that little tippy thing called an egglet here after doing this for so many years here. The egglet sort of shows up here. I'm just showing off my knowledge of string. In this example here, oh my God, my professor knows string, yeah. So anyways, the process of peristalsis is virtually identical to that. So in order to move things along, you have to have something that's going to be bunching it up and then pinching out behind it so you can stretch it back out. And so that's why you have to have 2 layers of smooth muscle. A circular layer that does like the pinching off, and a longitudinal layer that kind of does the bunching up. But it does it in such an extremely well coordinated effort that it just looks effortless. It doesn't look like you look when you're bunching. It just looks like one smooth motion. But yet it has to involve those 2 functions, a circular layer and a longitudinal layer, in order to move things along. We'll see there's another action that's called, I'll wait a minute. There's another action that's just going to mix food back and forth, but propulsion is the main one here. Now, mechanical breakdown. Mechanical breakdown, by the way, is primarily just breaking the very weak hydrogen bonds between adjacent molecules. We're really not breaking a lot of covalent bonds in this mechanical breakdown. Chewing, churning it in your stomach, for example, to break these bonds or break into smaller pieces. Segmentation, which is back and forth. And I have to also say that bile salts that are emulsifying fats also would kind of fall into that general category of mechanical digestion. But once you've broken it down so that the enzymes have surface area to work on, now we have true digestion, chemical digestion, which is going to involve breaking covalent bonds, using enzymes to do that. Those are catabolic reactions that are going to be breaking these very complex polymers down into their building blocks, into their monomers. You have to do this by the secretion of acids, by enzymes, buffers. All of those sort of things have to occur at the right place. And then once we have broken it down, we can absorb them, almost all in the small intestine. And then whatever is left over that you cannot digest, you remove by defecation. Removes the waste products in the form of fecal matter. This is sort of showing this, and we've already spoken about how we divide the organs of your digestive tract into two broad categories: alimentary canal,
Accessory.
also known as your food tube, and then your accessory organs. Alimentary canal from the mouth, oral cavity, pharynx, esophagus, stomach, small intestine, large intestine. Where the accessory organs are things like teeth, salivary glands, liver, gallbladder, pancreas. This is a nice little chart. I'm not going to spend much time, but it just sort of shows what different functions are going on in different regions of the small, large intestine, stomach, esophagus here. Worth mentioning a little bit of the peritoneum. Because as you've seen when you started your fetal pig dissection, the serous membranes that line the abdominal pelvic cavity are called the peritoneal membranes. They're the largest of your serous membranes in your body. And as serous membranes, as always, they will consist of two layers. An outer parietal layer would be the parietal peritoneum, and that's going to line the inner surface of your body wall. When you began your fetal pig dissection, you made those cuts around the umbilicus, right? You made the cuts down on here, and you open those flaps, right? Mm-hmm. Right? And you'll notice that the inside of those flaps, the skin on the inside, was shiny and smooth, wasn't it?
Mm-hmm. Exactly.
Why? What was that?
Perioral.
That's the peritoneal membrane there. That's the parietal peritoneal membrane that lines the inside of the body walls here. All right. The inner layer, the visceral layer, covers the surface of your internal organs, and in some areas of your intestines, they fold over itself to hold a lot of organs in place with a lot of blood vessels to form mesenteries. Mesenteries are just an elaborate extension of your peritoneal membranes. So there's mesenteries here, house a lot of blood vessels, lymphatics have to go through here. There's nerve inside of here as well, and helps to hold them in place. A couple of ones that are sort of mentioned here. This area here that's called the gray momentum usually sort of folds down over here. Sometimes you'll hear it referred to as the fatty apron, because they end up with a lot of fat deposits, abdominal fat deposits in that area there. The lesser omentum extends actually from stomach to liver, and nobody cares. But okay, histology of the anatomy. We have similar histological sections almost all the way from top to bottom. Basically a 4-layer system. We have a mucosa that lines the inside here that secretes mucus, and there can be digestive enzymes embedded within it here. All right. The mucosa ultimately has to absorb the products of digestion, right? Protect against infectious disease, hopefully by keeping pathogens from getting through. It's an epithelium here, which is mostly going to be simple columnar. When you did histology, well, nobody does histology here last semester because nobody was in here, but when you looked at your slides as simple columnar, almost every slide of simple columnar comes from the intestines. Mm-hmm. Because from one intestine, if you're a supply company like Carolina Biological Supply, from one intestine, you get like hundreds and hundreds of slides of simple columnar epithelium. Right? Now, you only have simple columnar epithelium basically in the central regions. All right? Top and bottom of your digestive tract, you're going to have stratified squamous to protect the top and the bottoms of your digestive tract here. So the very top and bottom, oral cavity, esophagus, that's going to be stratified squamous. You need that protection up here to protect you, for example, against hot pockets. Everybody understand that reference, I'm hoping? Good. And you need at the other end here for the anus, for example, to protect you, if nothing else, against like the really cheap toilet paper that they have here. I know people bring their own. I don't know where they get it from. I think they just get it from like the back of Home Depot. You know, it's just recycled sandpaper, I think. Seriously. It would flay the skin off your ass, people, if it were protected by stratified squamous epithelium. Oh, and that's in the faculty bathroom, people. I'm talking about the faculty bathroom. We don't even have like Charmin or like the Good Stuff or something like that. Okay, my issues, okay, moving on. Top and bottom, stratified squamous. Middle section's all simple columnar. And it's gonna be supported by the lamina propria, connective tissue below it. What kind of connective tissue would you expect? Areolar, because that's a given, isn't it? What's below, everybody, I've got a question for you. What would you find below the epithelial tissue? Let me try this again. What would you find supporting epithelial tissue?
Basement membrane.
What would you find below the epithelial tissue? Loose areolar connective tissue is the answer. You don't even have to know what kind of epithelium I'm talking about. What's below epithelial, everybody? What's below it? Areolar. What kind of connective tissue? Areolar. What's below connective, or I'm sorry, what's below epithelium? Areolar connective tissue. What's below epithelial tissue? Areolar. You're starting to get the, maybe you're starting to get the idea. Maybe you just think I need to be committed, which wouldn't be the first time. But I'm trying to highlight the fact that it's just a given under every epithelial barrier, there's going to be a supporting network of loose areolar connective tissue. It's just a given. All right. And this loose areolar is going to have, of course, all the things that you'd expect to have in it. A lot of capillaries are going to be in here. Mucus secreting glands here. There can be a little layer of smooth muscle. I would not ask you about that because it has very limited impact on really motility. The submucosa, which is right below it, is also areolar with a lot of nerve plexuses in them. Now this is worth mentioning because when we talk about the nervous system, we talk about your nervous system, basically brain, spinal cord, central nervous system. You have a semi-independent nervous system embedded in the walls of the digestive system. Now, that nervous system will communicate with your central nervous system, but it also works a little bit independently. It doesn't have a big functional brain, this nervous system of your digestive system, all right? It's a network of interlocking nerve fibers where there is no like central brain for it here. But nonetheless, this local nervous system is going to be involved in regulating a lot of the activities and coordinating some peristalsis and things like this. And we find some of that actually in the submucosa. We'll sort of see that later. But the submucosa is also where we're going to find the most important histological differences in the small intestine. Small intestine, if you have a histological slide and you want to know whether the slide that you're looking at is part of the duodenum or the ileum or the jejunum, you look in the submucosa because the mucosa is going to look relatively different. The muscularis, the muscle layer outside of it is, but the submucosa is where you find the regional differences. You'll see in the duodenum we have the submucosa there. We'll have mucus glands secreting mucus to protect the duodenum from all the stomach acid. When we get down to the ileum, the submucosa will have those Peyer's patches, those lymphatic nodules within the submucosa of the ileum to protect it against potential pathogens. The jejunum, the middle section, doesn't have anything special. That's how I kind of know you're in the jejunum. It's like, oh, there's nothing within the submucosa, must be jejunum. And we'll see that. So we have the submucosa, then we have the muscularis externum, which is the most important muscular layer. Notice that we have two layers of smooth muscle, an inner circular and an outer longitudinal to do that bunching and pinching thing. All right, and there's that nerve plexus. You know, sometimes they will call it the gut brain. If you ever come across that term, the gut brain, it's referring to this nerve network that's buried between these layers here. They call this nerve plexus the myenteric, myo referring to muscle, because it's going to be controlling the muscular contractions here. Okay. And in some places, this area, the smooth muscle is going to thicken, so we get sphincters. Sphincters are going to be obviously important for limiting movement from one section to the next while they're closed here. So it's the muscularis that's going to be responsible for peristalsis. And the other process here called segmentation is back and forth mixing. That's something that the small intestine really does. Because we want to be able to mix things along, get all the digestive enzymes going. So in the small intestine, most of what you're getting is this. Segmentation back and forth. With a little bit of peristalsis every once in a while, so we're slowly moving things forward. Anus that direction, slowly moving things in this direction. But most of it's segmentation. Back and forth mixing, also that muscularis is involved with that. And the outer layer, the serosa, connective tissue. Serosa, visceral peritoneum. Blood supply. Blood supply. Celiac trunk is the major branch off the aorta that's going to give blood supply to a lot of the digestive organs. Celiac trunk provides blood supply to the stomach, liver, gallbladder. Mesenteric arteries are the ones that are primarily going to be absorbing food. If there's a superior and an inferior one, which of them is probably going to be more involved in blood supply to the small intestine, the superior or the inferior one? What would your guess be? Well, which is probably higher up, the small intestine or large?
Small.
Small. So the superior is going to do more of small. So these plexuses that we talk about, like the myenteric plexus, this enteric nervous system, is this gut brain that we talked about, that communicate with each other and run a lot of sort of local reflexes here. There can be independence. They also can cooperate with the autonomic nervous system. And they're mostly the ones responsible for motility. We've talked about the two plexuses here. The submucosal nerve plexus, probably more involved in glandular secretions, because we're close to that. The myenteric plexus, more involved in controlling, regulating peristalsis, segmentation. But this brings us to two important terms. Two terms that are what? Important. And those are short reflexes versus long reflexes. Now, before going into detail about short versus long reflexes here, long reflexes are going to allow the autonomic nervous system to have an influence of digestive activities. Now, which branch of the autonomic nervous system will facilitate digestion? Sympathetic or parasympathetic? Parasympathetic. In fact, this is where the parasympathetic nervous system really shines. This is what it specializes in. This is why it's there, parasympathetic nervous system. Because it effortlessly can very carefully control all parts of digestive processes. And specifically, there is one parasympathetic nerve that will be involved in every parasympathetic effect from neck to navel. Which nerve is that? The vagus nerve. So when we talk about these long reflexes coming from the autonomic nervous system, the overwhelming majority of them are coming via the vagus nerve. All right? There are local sympathetic nerves that would shut down digestive activities, but to coordinate digestive activities, that's vagus nerve. Are there branches of the vagus nerve that control swallowing in your esophagus? Is the esophagus in your neck?
Yes.
Does vagus do every parasympathetic from neck to navel?
Yes.
Therefore, now when you begin swallowing, that's voluntary. When you begin swallowing,
Because you don't have to.
that's voluntary. But once it gets into my esophagus, it's going to be controlled by what nerve? Vagus nerve. And in fact, if you have difficulty swallowing, feels like things are always getting stuck halfway down, on rare occasions, that could actually be caused by problems with the part of the brain that controls what nerve? Vagus nerve. There'd be a lot of reasons for dysphagia, having difficulty swallowing, problems with the vagus nerve, only one of them. Short reflexes are reflexes done just within the plexus, within this enteric plexus, that doesn't need input from your brain at all. Short reflexes are just done by the gut brain itself, controlled entirely by the enteric nervous system. And these short reflexes are primarily going to be controlling those patterns of segmentation, peristalsis. Long reflexes are going to allow the autonomic nervous system to have an impact on it. It sends afferents to the central nervous system to receive input from both sympathetic and parasympathetic. Sympathetic is generally going to inhibit, where parasympathetic is going to enhance, increase digestive activities. And most of these long reflexes, most of the digestive, are also actually going to be coming from medulla too. Medulla not only is involved in control of respiration, but also some digestive activities, some digestive reflexes out of the medulla also. Like vomiting, the supermodel reflex comes out of the, did I say that? That was so wrong. They also comes out of the medulla. Keep in mind all these major things. Digestive activity provoked by a range of chemical, mechanical stimuli. There are receptors all throughout the walls of the digestive system that respond to distension, all right? When they are stretched, for example, all right? That respond to changes in the concentration of the fluids of the osmolar, even change in pH, substrates, so a wide variety of chemical stimuli can have an impact on it. The effectors of the digestive activity are limited to smooth muscle and glands. We don't control skeletal muscle via these local reflexes here. When stimulated, these receptors stimulate reflexes that make the smooth muscle contract to control peristalsis segmentation. They can also stimulate or inhibit glandular secretions. So for example, if you're seeing a seeing a commercial on TV for like Pizza Hut or something like this, and you see the steam rising off the pizza and all the gooey cheese being pulled like this. What happens to your mouth? If you are a normal human, you are what? Salivating here, right? Like Pavlov's dog. Doing that there. Is that increase in saliva a parasympathetic or a sympathetic? Parasympathetic. On the other hand, if you're really nervous, you're frightened, you're in a job interview, you're meeting the parents, the in-laws for the first time, that's what? Sympathetic. And then what happens to your mouth?
Dry mouth.
You get dry mouth because parasympathetic stimulates it where sympathetic is going to inhibit. Neurons and hormones are going to be controlling digestive activity, nervous system controlled by both short reflexes and long reflexes, but there's also going to be local acting hormones that you didn't touch on in 109 because these are not endocrine hormones mostly. Endocrine hormones generally have the potential for body-wide impact because they're released at the hormones that generally control digestive activities, they often will categorize as being paracrine, meaning that they're only going to affect local tissues, local activities, local acid secretion, for example, local amounts of local release of digestive juices from the gallbladder or from the pancreas, controlled by these local acting hormones here. Again, the types of motility issues, peristalsis is directional. Involves, again, as we mentioned, the coordinating contraction of both the circular layers and the longitudinal layers for that one here. Segmentation, just moving things back and forth. You just need that little circular layer to sort of mix things around. You're not going to be pushing things along. So segmentation, you're just doing this circular layer to mix things back and forth. That's segmentation here. It is not directional. Segmentation just goes back and forth. So take a look at what's happening here. We start by ingesting food in the oral cavity. We begin to mechanically break down food by the process of mastication, which is, again, is a word you have to be very careful with. As I've sort of said before, if you are eating pizza up in your bedroom, people, you do not want to be do not want to be yelling down to your parents, "Don't bother me right now, I'm busy masticating," because they're not going to understand, they're going to call the priest or something, so careful with that word. We're going to involve mechanical breakdown, we initiate process, limited digestion of polysaccharides because amylase, salivary amylase is going to be released into the oral cavity. Almost nothing gets absorbed in your oral cavity. Very little can get absorbed in the oral cavity. Some rare drugs because there's a membrane, that little disgusting membrane under your tongue, the frenulum. You know what I'm talking about? That's a thin enough membrane that you can absorb some, especially things that are lipid soluble, like nitro. Alcohol is so, almost nothing gets absorbed until you get to the intestines. But alcohol is so lipid soluble, it can start to be absorbed even in your mouth and even on the way down. That's why you can get hammered so quickly if you put your mind to it. Not that anybody would do that, but I'm just sharing that with you because alcohol is just so lipid soluble. It's even in being absorbed in the oral cavity at least a little bit. Oral cavity, whatever, hard palate, soft palate, nothing with this. Nothing about the tongue that I'm going to ask you about or intrinsic, extrinsic, there's nothing there. I will ask you some questions about teeth though. I'm not going to ask anything about the tongue, but I am going to ask about teeth. Keith brings up the fact that we are omnivores. What is the difference between a carnivore, an herbivore, and an omnivore?
Meat, veggie, both.
Did everybody catch all that? Carnivores consume
Meat,
almost exclusively meat, where herbivores
plant-based.
just plant, omnivores
Both.
can do both, right? Now the reason for that again is that if you are a carnivore, for example, you can pretty much get all your nutritional needs met just from eating meat or whatever's in that. If you're an herbivore, you can get all your nutritional needs met just by eating plant matter. If you're an omnivore, it usually means you need to have some of each in order to survive. Not that you can, but generally that you must eat some combination and we fall into that category of being an omnivore. There's some things like vitamin B12 that you just really have to get from animal sources. You just don't get really vitamin B12 from other sources. If you are a vegan, you need to make sure that you're getting supplementation of vitamin B12 or you're going to suffer pernicious anemia. I think we had that talk earlier here. You need vitamin C, for example, will only get from plant sources. Just one example. Have we had that talk about vitamin C in here before? No? Okay. If you don't have vitamin C, if you don't get vitamin C, is that a good thing, bad thing? Is there a disease state associated with not getting enough vitamin C? Scurvy. And scurvy can kill, all right? They didn't know for the longest time actually what caused scurvy. It wasn't really until the last century that they were to trace it down and figure out, that was a vitamin C issue, all right? And you need it from plant sources, all right? They realized that, the people that would get, little history, people that would tend to get scurvy in the past generally fell into two big categories. They don't know why, but the two big categories were people that were sailors, for example, or going on long voyages here. They tend to get scurvy. And the other group that tended to get scurvy were pilgrims who are making religious pilgrimage. And the reason for that is that generally those individuals did not carry fresh food with them. It was just too heavy to carry that along with them, so they carried dried things along with them or dried meats for this, and very little fresh fruits or vegetables, which is where you get vitamin C from. Now, somebody figured out that, at least in the British Navy, that limes were a source of that would keep for a while, which is why they would call the British sailors limeys for a while, without realizing it was the vitamin C that was the issue behind it here. But your cat, even though it will never eat any vegetable or drink any orange juice, will never get scurvy. Your dog will never get scurvy. If you have a pet mouse at home, pet rat, it's never going to get scurvy, even if it never eats any vegetables. Why? Because they can all make it themselves. Most mammals have the gene for making their own vitamin C. We do not. We have a broken gene. They call it a pseudogene. And we share that with all the primates. All the higher primates that are related to have that same broken gene, where we still have the gene in there, but it's mutated in such a way that gene cannot be expressed, which is why we have to get vitamin C from our diet. Now, I'm bringing this all up at this point in time now because our dentition pattern, our teeth, reflect the fact that we are an omnivore, because we have some teeth that are really more carnivore teeth, and we have other teeth that are more herbivore teeth. So what are your real carnivore teeth? Yeah, your fangs, right. The canine teeth, the cuspids here, the canine teeth. Where are we showing here? Here's our canines here, all right, your fangs, all right? Because they're good for like ripping meat from the bone, right? You got that tough steak on the side here, you're gonna sink your fangs into them. Some people actually are self-conscious about like the length of their canine teeth because they think they make them look a little bit too vampire-like, and will actually have them filed down for cosmetic purposes so that they don't look like a vampire going out there, right?
What? I want mine sharpened.
You just want your sharpen, you want them
Yeah.
sharpened into points? Get the
Yeah.
sharpened points.
It's really.
So you look like a shark, okay, yeah.
No.
That's a scary woman.
Just the king size.
What kind of teeth do we have that are more related to the fact that we need vegetable matter, that are more herbivore teeth? Well, a couple of them. First of all, the ones in the front here, the incisors, all right, are good for like clipping and cutting food because they sort of go each other this way. This is what a horse will be doing when they're clipping grass right there. They're using what teeth in the front there? Their incisors to do that. And aside from the incisors, the other big ones are the molars, as people mentioned. The molars